The overall goal of this procedure is to use a method known as Tandem Affinity Purification to isolate protein binding partners. The TAP tag used in this study is an N-terminal tag consisting of 2 units of protein G and a streptavidin binding peptide. These are separated by a TEV protease cleavage sequence that allows specific elution. The bait protein, in this example murine EIF4E, is at the C terminus of this fusion construct. The TAP tagging protocol is a 6-step procedure. After transfection and generation of cell lines, cells are induced to express the tagged bait protein before being lysed in a mild lysis buffer. 2 affinity purification steps and 2 specific elutions are used to purify the bait and any potential interacting partners. To generate cell lines, HEC293 cells should be transfected with the pMEP4 expression vector encoding the TAP-tagged bait protein. Cells are then selected with 100 micrograms per ml hygromycin B. Since the plasmid is maintained episomally, there is no need to isolate specific clones. 10 confluent flasks of cells are induced by treatment with 10 micromolar cadmium chloride for 16 hours before being scraped off into media. The suspended cells are then transferred to 50 ml tubes and the cells pelleted by centrifugation. These cells are then washed 3 times with ice-cold PBS before finally being pelleted. Step 2. Cell lysis. Cells are lysed in 5 ml of cold lysis buffer by repeated pipetting before being left on ice for 5 minutes. To ensure efficient lysis, the cells are then syringed through a narrow gauge blunt needle and freeze-thawed. The resultant lysate is alicotted into 1.5 ml tubes and unlysed cells and debris removed by centrifugation. Of note, the pellet size is markedly reduced following this centrifugation step. The lysate supernatants are then combined in a 15 ml falcon tube and passed through a 0.45 micrometer filter to remove any additional debris. A 50 microliter aliquot of this lysate should be taken for subsequent analysis. This sample is referred to as sample 1. Step 3. Binding to rabbit IgG agarose. In this stage of the protocol, the protein G tags are immunoprecipitated by rabbit IgG agarose. To alicot agarose beads, remove the end of a pipette tip. This helps to avoid damaging the beads. Gently resuspend the rabbit IgG agarose solution by swirling the bottle, before removing the agarose to a 15 ml tube. Wash the agarose 3 times in chilled lysis buffer using low-speed centrifugation to clarify the beads at each step. The pelleted rabbit IgG beads should be visible after centrifugation. After each step, carefully remove the buffer without disturbing the beads. After the final wash is complete, add the filtered and clarified lysate to the packed beads and incubate for 3 hours, or overnight if preferred, at 4 degrees C using a rotating mixer. Following the immunoprecipitation, spin down the agarose beads and remove the supernatant for subsequent analysis. If required, you can use a fine billed tip to remove any remaining liquid from this tube. This supernatant is sample 2. Step 4. TEV protease cleavage. In this step, the bait is cleaved from the protein G tags. This step allows specific cleavage of the bait protein, effectively an elution step from the rabbit IgG agarose immunoprecipitation. Prepare the TEV cleavage mix and add this to the agarose beads. Using a pipette tip with its end removed, transfer this mixture to a siliconized microcentrifuge tube and invert to mix the agarose into suspension. Prior to overnight incubation at 4 degrees C, remove an aliquot of this TeV cleavage reaction for analysis. This is sample 3. Overnight incubation on the rotating mixer should allow the TeV reaction to progress to near completion. Step 5. Binding to immobilized streptavidin beads. In this step, the cleaved bait and any potential binding partners are affinity purified from solution. Firstly, centrifuge the TeV cleavage reaction containing the rabbit IgG agarose beads to pellet the agarose. Remove an aliquot of the clarified supernatant for analysis. This is sample 4. Carefully remove all the remaining supernatant, which contains your bait protein, to a fresh tube. Again, use fine billed pipette tips to remove all possible supernatant. Retain the rabbit IgG agarose beads for subsequent analysis. This is sample 5. To prevent damage to the streptavidin beads, remove the end from a pipette tip. Gently resuspend the beads and remove an aliquot to a siliconized tube. Wash the streptavidin beads with lysis buffer, clarifying by low-speed centrifugation after each wash. After each wash, carefully remove the buffer. Streptavidin beads are very small and can be easily displaced, even when using fine-billed pipette tips. After the streptavidin beads have been washed, add the supernatant from the TeV cleavage reaction to these beads and incubate the reaction for 3 hours or overnight at 4 degrees C using a rotating mixer. After incubation, centrifuge the streptavidin beads and remove the supernatant. This is sample 6. Of note, the bait protein is now associated with the remaining beads in the tube. Wash the streptavidin beads containing your bait protein with lysis buffer to remove any additional contaminating proteins. Remove the remaining wash solution from the beads and leave them on ice. Step 6. Biotin elution. In this step, biotin is added to the streptavidin beads. The interaction between biotin and the streptavidin beads displaces the bait, which goes back into solution. Add the biotin solution directly to the beads and invert to mix. Incubate the reaction at 4 degrees C for 3 hours or overnight using a rotating mixer. Centrifuge the biotin agarose mixture and carefully collect the mixture into a fresh tube. This is your final eluate, referred to as sample 7. The remaining streptavidin beads are sample 8. Because of its low protein concentration, this final eluate needs to be concentrated prior to analysis. This is achieved using low molecular weight cut-off spin columns. Reduce the volume by centrifugation, monitoring the process regularly. This concentrated sample can be split and analysed on SDS page gels by Kumasi and Silverstein. If the samples are to be analysed by mass spec, we advise using precast commercial gels. Individual bands can then be extracted and the proteins identified by mass spec. In this case, the bait was murine EIF4E protein.